Alright, so now that we've taken a look at the taxonomy and the classification of viruses and diseases, viral diseases, and then taking a look at the viruses themselves, their structure, which is quite simple, we're going to take a look at the viral life cycle. So there are two different life cycles in a virus, and we'll take a close look at both of them. First, let's just take a close mention that, or let's mention here that we are going to be looking at bacteriophages or bacteriophages. They are a good model, as it mentions here, for understanding how viruses affect the cells that they infect, because we see the same types of processes in eukaryotic viruses, so viruses that infect humans, for example, and other eukaryotic organisms. So what we see is that we have what are called virulent phages, which typically lead to the death of the cell through lysis. Lysis is bursting or breaking open of the cell, breaking it apart. And then we have temperate phages, and those can become a part of the host chromosome and then are replicated within the cell's genome, and then they'll stay in there until they are induced, and then they make newly assembled viruses, or what are called progeny viruses. So let's take a look at the first cycle, which is the lytic cycle. So during the lytic cycle of a virulent phage, the bacteriophage takes over the cell. It reproduces new phages, and then it destroys the cell. So the lytic cycle is going to be this process down here. There are actually 5 steps to this process of the cell, or of the virus rather, taking over the cell, making copies of itself, and then destroying the cell. We have the T-even phage is a good example of this, of a well-characterized class of virulent phages. There are 5 stages that we're going to take a look at. So the first stage, I'll just scroll down just a bit here. The first stage here is called attachment. So we have our bacteria, and then, so remember we're talking about bacteria cell here. So this is our bacteria cell and our bacteriophage, which is a virus that infects bacteria. And what we see first is we have this attachment. So the bacteriophage is going to attach itself to the bacterium. So it attaches it to the surface of the host. And the way that it does that is that it's going to bind with these specific bacterial surface receptors. So some of these lipopolysaccharides, if you remember back to our different types of bacteria, or other different proteins that are on the surface of the cell. So most of these phages, just like other viruses, have a very narrow host range. Remember, the host range, or the number of different types of cells or organisms that it can infect. So they, again, are often very narrow. Specifically, bacteriophages are very narrow and may infect only one species of bacteria, or even one strain within a species. So this actually ends up, they mention here that this can be exploited for targeted treatment of bacterial infection by phage therapy or by phage typing. And we're going to take a closer look at those kinds of things toward the end of the chapter. So the first stage is attachment. We find that on the bacterial cell there will be little proteins, and then this specific bacteriophage can see that particular protein, attach to that protein, and then move on to the second stage, which is penetration. And in this stage, this is where the bacteria, or this is where the virus is going to get its DNA or RNA into the bacterial cell. So the viral DNA enters the host cell. So you can see here, during this stage, we can look at the words up here. This occurs through contraction of the tail sheath. So remember that there is this sheath on here. It contracts, and then it acts like a hypodermic needle. So it sticks this middle part here, this helical piece of it, and it is going to stick it through the wall of the cell here, through our outer membrane, and then it's going to inject that viral genome through the cell wall and the membrane and get it into the inside of the cell, into the cytoplasm. So the phage head and the remaining components remain outside of the bacteria. So you can see here, all of this stuff is going to stay outside of the bacteria. All that's going in is our phage DNA. The pink here is our host DNA. Then we have step 3, which is biosynthesis. I'll scroll down here so we can see the words as well. So the third stage is biosynthesis, and this is where it's going to make these new viral components. So after it's entered the cell, the virus is going to synthesize virus-encoded endonucleases. Aces is enzyme, and then endo meaning inside, the nucleus aces. So what that's going to do is it's going to degrade the bacterial chromosome. So part of this lysis, the lytic cycle, is that we are going to attach to the cell, we're going to get the phage DNA into the cell, and then during biosynthesis, the actual genome that was put inside of the bacteria has the components to synthesize these enzymes, these endonucleases, that then go to the bacterial chromosome and just degrade it, bake it, cut it up into pieces and degrade it and get rid of it, so that then it can hijack the host cell, and then it can replicate, transcribe, and translate all of its necessary viral components. So this, remember, the pink here is our host DNA, and you don't see any of the pink over here because it has completely broken it apart and degraded it. And so then it can take over all of the other mechanisms within the bacteria cells. So all of the things that make proteins, it can use the ribosomes and everything that it can to go through this replication, transcription, and translation in order to make its capsomeres, which were the proteins to make its capsid, the sheath, base plates, tail fibers, and other viral enzymes. So it'll go in and it'll get rid of its genome, the bacterial genome, so that it can take over and do what it wants to in the cell to make more copies of itself. So then it assembles new viruses. It does mention here that polymerase genes, so remember polymerase is so that we can go through transcription and translation, are usually expressed early in the cycle, whereas the capsid and the tail proteins and things are expressed later. So it'll go through the process of making components or making proteins that are necessary to then do things further on in the process to make more pieces of itself. Then the next one, step 4, is maturation. So then during maturation phase, we have our new virions created. So in step 3, we make all the components, and then in step 4 is when they take all those components and stick them together the way they're supposed to be to make new viruses. So to liberate these free phages, then the bacterial cell wall is going to be disrupted by phage proteins, such as holin or lysozyme. So they're going to break apart this outer portion, and then this is step 5. We have lysis and release of these new viruses, so these bacteriophages or bacteriophages. So we put them together in the maturation phase, and then in step 5, the cell lyses and it releases all of these newly made phages. So again, lysis is this process of bursting or breaking the cell, and then what we release are called progeny viruses. Okay, and then they can go out and then they can infect new cells. So these are the 5 steps to our lytic cycle. So this is when they're virulent. Okay, now what we're going to do is we'll take a look at the other cycle, which is the lysogenic cycle. In the lysogenic cycle, we still see that this phage genome is going to enter the cell through those first 2 parts. The attachment, it's going to attach to a particular protein that it recognizes. Remember, it has a host range, so it'll attach to a specific protein, and that has penetration, so it'll still stick that sheath part like a hypodermic needle in there and then inject its DNA. An example of this is the lambda phage. So the difference then is that if we look up here, so we just said that we have the 2 pieces that are the same. Is step 1, the attachment part, so we have the bacteriophage attached to the bacteria cell. And then we have penetration, which is getting its phage DNA inside of the cell. Then we see a difference. Okay, at this point, during the lysogenic cycle, instead of killing the host, so remember we made those endonucleases in the lytic cycle that went in and then degraded and broke apart the host cell DNA. Instead, what we see is that the phage genome integrates itself into the bacterial chromosome, and then it becomes part of the host cell, part of the host genome. So it doesn't break it down. Instead, it goes in and makes a very precise cut in the genome, in the DNA, and then it puts in its own DNA. The integrated phage genome is called a prophage. So when it is in there, so when we've put the piece of viral DNA into the host DNA, that is then called a prophage, that DNA inside of the host DNA. Then a bacteria cell that has that happen to it is called a lysogen. So a bacteria that has been infected with a bacteriophage and has a prophage in its chromosome is called a lysogen. So then the process in which a bacterium is infected by a temperate phage is called lysogeny. Okay, so what we're describing here in the lysogenic cycle is what it looks like for a cell to have undergone lysogeny. So it is typical of temperate phages to be latent or inactive within the cell. So at this point, so we have attached, our bacteriophage has attached to the bacteria cell, it has injected its DNA into the cell, and then it has integrated its DNA into the host DNA. And then at this point, it can be latent or inactive. It's not going through a process of actively making copies of itself to then burst out. So in this process, the bacterium is going to replicate its chromosome, and then in the process of the bacteria replicating its own chromosome, it also replicates the phage's DNA. So since it's already in the chromosome, it's going to replicate that as well, and then that's going to pass it on to new daughter cells during reproduction. So when the bacteria is going to make an exact copy of itself by binary fission, remember that's what happens when a bacteria cell separates and makes a new bacteria cell, they're exactly the same. They're going to copy their DNA so that they can put it into the other bacteria cell. When it does that, it's going to also copy the phage's DNA because it's within its own chromosome. And then when it splits off into the bacteria or into the daughter cell now already has that DNA in it. So they do note here that the presence of the phage may alter the phenotype of the bacterium because the phage can bring in extra genes. So it brings in other genes like toxic genes, so toxin genes that can then increase bacterial virulence. So this change in the host phenotype, so when a bacteria changes its phenotype, changes what it expresses, due to a phage being in it, which again is called a prophage, and when the bacteria cell has a prophage in it, it's called a lysogen. So when that happens, it's called lysogenic conversion or phage conversion. So we have had this conversion or this change due to the fact that it has been infected by a temperate phage. And then it gives some examples here. The Vibrio cholera and Clostridium botulinum are less virulent in the absence of the prophage. So when we're talking about Vibrio cholera or Clostridium botulinum, they're not as difficult to deal with, difficult to handle. They're not as virulent unless they have this prophage in them. So the phages that infect these bacteria carry the toxin genes in their genome and then enhance the virulence of the host when these toxin genes are expressed. So in the case of Vibrio cholera, the phage-encoded toxin can cause severe diarrhea. So whereas before it may not cause diarrhea, in the presence of a prophage, it would. In C. botulinum, the toxin can cause paralysis. So the phage itself is what has that toxin coated in it, and then once it has injected itself, integrated itself into that bacterial genome, then it can cause paralysis. During lysogeny, the prophage will persist in the host chromosome until induction. Okay, so the prophage, right, which is the DNA that's integrated itself into the chromosome, will just hang out in there, and it will just keep being copied and then put into new daughter cells over and over and over again until induction. Induction is when it's going to excise itself, take itself out of the chromosome of the bacteria cell, and then it's going to go into the other stage. Okay, back into the lytic stage. So let's take a look at that in images here. So here we have the phage infects the cell. Just like we said before, there's attachment and then penetration, which is where we see the DNA going into the cell. Unlike the lytic stage, we have in our lysogenic stage, the phage DNA is going to become incorporated into the host genome. And so they have the phage DNA as the blue there, and then the pink is the bacterial genome. Then, as we mentioned, the cell will divide, and then in this case now we have the prophage DNA is passed on to daughter cells. So anytime it divides, it's going to copy the chromosome, including the prophage, and then that will go into all the daughter cells. Then, once we get into a situation where things get stressful, so maybe there's not as much food around, maybe the temperature changes, things like that, then the prophage DNA is going to cut itself out, excise itself from the bacterial chromosome. At this point, it's going to enter the lytic cycle then, which is just like the cycle we already talked about. So at this point then, the DNA is going to replicate itself, and then it's going to make all the different pieces that are needed, so the capsid and the sheath and the tail fibers, all of those different parts. And then it's going to assemble them. The new particles are going to assemble into new virions, and then the cell will lyse, releasing these newly made phages. So the lysogenic cycle is this first part here, and then we can move into the lytic cycle after that. And this is with a temperate bacteriophage. Okay, so a virulent bacteriophage is going to simply go into the lytic cycle. When we're talking about a temperate bacteriophage, it'll go through the lysogenic cycle, and then when things get a little difficult, it's going to go into the lytic cycle and then release copies of itself. It says, okay, no longer time to be dormant. We're going to release copies of ourselves and make sure that we can continue our, quote, life cycle, and then go out and make more copies and infect more cells. All right, so then another important thing that has to do with these viruses and infecting of bacterial cells is seen in transduction. So transduction occurs when a bacteriophage transfers bacterial DNA from one bacterium to another during sequential infections. Okay, so there are two different types of transduction, and we'll look at these a little bit closer. So there's generalized transduction and specialized transduction. So during the lytic cycle, okay, this is when it's virulent, during the lytic cycle of viral replication, again, the virus is going to hijack the host cell, so it's going to take over its machinery so it can make copies of itself. Remember, it also degrades the host genome, or degrades the host chromosome, and then makes more viral genomes. As it goes through this process, as it is going to assemble and package up the DNA into its own phage head so that it can burst out of the cell and infect other cells, that packaging occasionally makes a mistake. So instead of packaging up only viral DNA, what ends up happening is it's going to take a random piece of host DNA and then insert it into the capsid along with the phage DNA. So it will accidentally pick up a little chunk that was cut apart but is still floating around, and then it packages it up inside of its capsid with the phage DNA. And then once this is released, so then it's going to go through the same part of the lytic process. It will break out of the cell and be released. This virion will then inject the former host's DNA into a newly infected host. So it bursts out at the end of the lytic cycle. Then it's going to find a new cell. It's going to go through the same process, go through the same lytic cycle. It's going to attach itself, it's going to penetrate, inject the DNA into the new cell, and when it does that, rather than just injecting the virus genome, now it's going to be injecting the former host's DNA as well, that little tiny piece that it accidentally picked up. So then this genetic information can allow for DNA recombination to occur. And then what happens is that the new host is going to get new genes from this. And this is seen, for example, in antibiotic resistance. So on accident, a virus, a bacteriophage, may pick up some sort of antibiotic resistance gene from one bacteria, and then through the process of infecting a new bacteria, it gives it that antibiotic resistance. So let's just take a look down here really quick at the image of what we just talked about. So we have the first steps that we already know, viral attachment and penetration, we get that in there. Then we have integration. Oh, actually, this is for the second one. Hold on. All right, so generalized transduction. Generalized transduction occurs when a random piece of bacterial chromosomal DNA is transferred by the phage during the lytic cycle. All right, so that's what we had just talked about. Sorry, I thought that's what this image was, but it's not. So generalized transduction is what we said. There's a random piece of DNA that just happened to get caught up in the process and get put inside of the capsid, and then is sent along, and then it goes into the next bacteria. Then we have specialized transduction. In specialized transduction, this is when it happens at the end of the lysogenic cycle. This is when the prophage is excised, and then the bacteriophage enters the lytic cycle. So remember, in our lysogenic cycle, we have the prophage, which is basically just the bacteriophage's DNA is now inside of the bacterial chromosome. And then at the end of the lysogenic cycle, we have induction occur, which is when we're going to cut that out. The bacteria, or the virus is going to cut out its DNA. And what we see here in specialized transduction is that when it does that, when it goes in and it cuts out that DNA, it accidentally cuts out genes on either side of its own DNA. So it may cut out more on one side of its DNA, it may cut out more genes or more genetic information on the other side of its DNA, or both. So then this is called specialized transduction. So when it cuts out more than just itself, then what happens is that can be transferred to the next one. So since the phage is integrated into the host genome, the prophage can then replicate as part of the host. However, some conditions, like ultraviolet light exposure or chemical exposure, are going to stimulate the prophage to undergo induction, right? So we're going to go into the induction part, causing the phage to excise from the genome and then enter the lytic cycle. So this is where it goes through the process of describing that while it's going through excision from the host genome, it may occasionally remove some bacterial DNA near the site of that integration, so on either side. So then the phage and the host DNA from one end or both ends of the integration site are packed within the capsid and then are transferred to the new infected host. So since the DNA transferred by the phage is not randomly packaged, but is instead a specific piece of DNA near the site of integration, this is called specialized transduction. The DNA can then recombine with the host chromosome, and then it gives the latter new characteristics. So we see the same thing here, where if we take a piece of genome, a piece of DNA from one bacteria on either side of our viral genome, and then give it to a new cell, we can be transferring genes like, for example, antibiotic resistance to the other bacterium. And they mentioned that transduction seems to play an important role in the evolutionary process of bacteria, giving them a mechanism for asexual exchange of genetic information. So we can see bacteria changing over time based on this process happening here, based on the phages going through what they are going through for their own, quote, life process, and then that changes bacteria as well. So now down here, this is our specialized transduction, as mentioned here. We have our viral attachment and penetration, and then it integrates itself into the cell. So you can see the blue genome there is the viral genome, and then the pink is the bacterial genome. And then we have excision. So when the phage, which is the blue, is excised from this bacterial genome, it accidentally takes a short piece of the bacterial genome with it. And then that is going to be packaged up inside of our virions or virions, and then when it goes along, when these then break out of the cell and go into a new bacteria cell and inject the DNA during this stage here, but in a new cell, it's going to take, you can see, and put a little piece of that original host, this guy's chromosome, into the new bacteria here. And then they're showing here recombination. So the phage DNA along with the attached bacterial DNA are then going to be incorporated into this other bacteria's genome. And then that's what they're showing here with this lighter colored blue is the DNA from the original bacteria going into the second bacterium here. And then that could be, for example, antibiotic resistance that then is given to another bacterium.